Okay, good morning. Welcome to the 15th webinar series, Structure Meets Function. This time, this webinar will be from the Portuguese participation at Instruct. And I'm sure that all of you know what is Instruct-ERIC, which is a European Research Infrastructure Consortium for Structural Biology Research. And the main activities within Instruct are research visits, training, internships, and small pilot research and development funding projects. Instruct has started as a pan-European infrastructure. Can I have a second slide, please? Has started many years ago, it became an ERIC in 2012, if I'm not wrong, and now it has 15 member states, and each one pays a subscription that allows the scientists to access the range of services and high-end technology platforms that are available through their centers. Portugal was fortunate to be one of the first countries starting a founding member of Instruct, but many more have subsequently became members. And now, as I said, we are now 15 members. The access to the facilities, which is the main activity of Instruct, is distributed through 11 centers and through 26 facilities across Europe within these centers. I should also tell you that this year, Instruct opened a call for Instruct sites. Instruct sites are supposed to be kind of organization between the non-center and a center. So are for where this call was directed to the members which do not have a center. And this call was very successful. The Instruct sites will be mainly the main mission for Instruct sites will be participate in training activities. There is no funding for the Instruct sites as such, but they will have to present proposals within the open calls as usual, and being evaluated, and if funded, will go ahead with their training activities. Portugal has applied with 2 independent proposals, which are then suggested to combine in a single proposal, a single site. And we are now working on that. Margaret Archer from ITQB was the proponent of one of the proposals, and Carlos Cordeiro from FC, the Faculty of Sciences, the University of Lisbon, was the other. And they are now working together with Instruct Hub to prepare the presentation of the Portuguese Instruct site at the Instruct webpage. We will now start with our speakers of today. Carlos Cordeiro, as I mentioned already, is from Faculty of Science of the University of Lisbon. So, Carlos Cordeiro is our expert within the Portuguese network of users of Instruct on mass spectrometry, and he has recently acquired at the faculty a new instrument on magnetic resonance mass spectrometry, and he will tell us about breaking the limits of native mass spectrometry with this technique. Thank you very much, Carlos. Go ahead, please.
Thank you very much, Arménia. Good morning, everyone. It's a pleasure to be with you all, even if it's on a virtual setup. So native mass spectrometry is a technique that allows us to measure the mass of proteins and protein complexes. And right, so it allows us to measure the protein mass of complex. So we can determine stoichiometry, we can determine composition, interactions with small molecules, interactions between proteins, screening. So it's a very useful technique. It does not provide atomic coordinates, of course, but it provides important information concerning stability, topology of binding, and information obtained by native mass spectrometry can be very useful in guiding further studies. But why do we need to break the limits of native mass spectrometry with magnetic resonance mass spectrometry? The main reason is because we need resolution. We need to be able to see things. If you look at this monument, like if it was a protein complex, you could see that it has a lot of components, and including some, I would say, small molecules attached to it, like this small ship that you can barely see in the hand of this statue. If you are working with low resolution instruments, that means lower than 10,000, basically you have a blurred image. You see there is something there. You see a general shape, but you don't see exactly detail. If you go further in resolution, say 10-fold higher, up to 100,000, now you are in the realm of high resolution. And this is accessible to quite a lot of modern instruments. And now you can see details, but still you don't see the fine details of reality. And in many situations, this is what you want to see. For that, you need to go to extreme resolution. So 10-fold higher, and you get 1 million resolution. And of course, now you can see the fine details of this caravel. And with these fine details, you can build a detailed model that is more accurate to your reality than if you are seeing something like this. Now, a few mass spectrometers can achieve 1 million at low m/z values, which are not very useful for native mass spectrometry. And only a handful of instruments, magnetic resonance mass spectrometers, also known as Fourier transform ion cyclotron resonance mass spectrometries, can actually go beyond this limitation and reach 10 millions or even 100 millions. It has been recently demonstrated that it's possible to reach something like 146 million. So can we reach these levels of resolution on native mass spectrometry and use this tool? Well, the benefits of resolution are also very obvious at the molecular realm, because if you are working with very, very complex mixtures, you want to separate the components. So in this case, you have an extract of yeast. So these are small molecules. Basically, you see a very complex spectra with thousands of peaks, but you can make sense of these peaks because you can see there are patterns, and these patterns have certain differences, different heights. So you can actually relate these patterns with the atomic composition of individual molecules. So you can actually separate the isotopologues and immediately obtain accurate molecular formulas. For native mass spectrometry, we also need resolution. So this is a small tetrameric protein. And as you can see, you determined mass accurately within, say, less than 10 Dalton at a resolution that is about 10 to 50,000 to 20,000, not more than that. And basically, from the charge state distribution, you can calculate a very accurate mass. This difference would be enough to detect post-translational modifications or the binding of small molecules. But now, in the same m/z range, for a completely different protein, which has a much higher molecular mass, but not so much higher, which is actually the trimeric spike from SARS-CoV, you see a completely different picture. Basically, we can see the monomer, and you can see the trimeric form, but you do not see resolved charge states. You see some glimpses of a possible charge state distribution. So you cannot accurately calculate the mass of this molecule. And this happens because it's quite heavily modified, so it's highly heterogeneous. So it produces a very broad charge state distribution with a lot of overlapping peaks, which is preventing us from calculating an accurate mass. We can still relate the masses of this. We can still study the transition from one form to the other. So we can still monitor the formation of the trimeric form, but definitely not within 10 Dalton. So we want something better. And this is the path we chose to follow. So what limits the resolution in native mass spectrometry has been investigated by several groups. And basically we can resume this to 3 points. So one is the natural isotope distribution, which, so as molecules become more and more complex, larger, or heterogeneous, of course, you get broader envelopes and overlapping charge states and isotope log. So this is inherent to your sample. And as much as you work, it's very hard, if not, I would say impossible, to overcome this limitation. So the other one is dissolvation. When you place a protein or protein complex in the gas phase, it usually flies with some solvent, salts, adducts, and a lot of other things that adds to this heterogeneity. So proper dissolvation is crucial to obtain a good resolution mass spectrum. And this has nothing to do with most of the instrument, your instrument, only with your ion source. And of course, you are limited intrinsically by the resolution of the mass analyzer, which in many cases, you do not notice in native mass spectrometry, because until now, I would say, most of the research that has been done in this field actually carries about large mass variations. So you can get away with a relatively, I would say, low resolution, less than 100,000, easily to do most of your research. So what do we have to do to go further? So traditionally, native mass spectrometry has been done in QTOF instruments, usually modified and sometimes quite heavily modified. So those modifications happen in the source region by carefully controlling the pressure, not only in this area of the source, but also in the collision cell, so to stabilize non-covalent aggregates. The quadrupole and also these multipoles, the frequencies that are associated to it, and the range of frequencies that you have access allows this kind of quadrupoles to isolate ions, say, of 30,000 or more. Usually on a normal instrument, you cannot go above 2,000, or in some situations 4,000 or maybe 6,000, but not in the tens of thousands, which is good, very good if you want to isolate and fragment your molecule in the gas phase. The pusher is also modified for low frequency. And in general, this has been the mainstay of most, if not a large fraction, a vast majority of studies in native mass spectrometry. So more recently, another instrument has been introduced, and this is a Q-exactive type Orbitrap, but also highly modified. So I think it's called the extended mass range or whatever, because the nomenclature changes a lot, and this is in constant change. Quadrupole is also modified. And of course, you have a much higher resolution analyzer. And in some ways, because of the specific ion optics, you also get a very good dissolvation. So the combination of these factors renders this kind of modified Orbitraps most likely the highest resolving instruments for native mass spectrometry presently. So there are already quite a few installations, and it's quite predictable that this will keep increasing. But of course, they are limited in the inherent resolution of the Orbitrap, as the Orbitrap analyzers that are coupled here usually do not reach 1 million. They are usually lower than that. So all in all, they are intrinsically limited by this. The other instrument that has been used by some laboratories, and I would say not many groups, is magnetic resonance, or FTIC-RMS. Although this was invented in the 60s, and the first commercial instrument, I think, was made by Nicolet in 1976. Only recently, some serious modifications in the source region, and especially the introduction of a new cell, allow us to translate the magnetic resonance phenomenon in something that is extreme resolution, easily exceeding 1 to 10 millions in most installations. Of course, these instruments are quite rare. I think there are less FTICRs in Europe than there are CryoEM microscopes, for example, even high resolution ones. So a group of researchers working in this area created a consortium that is funded by the European Union to allow other researchers to have competitive access to our instruments. So we have 10 laboratories in Europe, and actually native mass spectrometry work is mainly done in one of the pioneer groups that use the FTICR for this in southern Finland, which is led by Jan Janis, and also in the Czech Republic by Petr Novak. So as you can see, we have a good coverage of Europe, although the main density of instruments is around the Central Europe. So magnetic field is important, and usually it varies, I would say, between 4.7 and 21. There are only two 21 Tesla instruments in the world. In the United States, so the largest field in Europe is 15 Tesla, but there are plans to install 18 Tesla in one of the consortium laboratories. Of course, magnetic field, like in NMR, imparts on the performance of these instruments, but it's not as critical as in NMR, as most of the figures of merit actually vary linearly and not quadratically or on another power. For example, resolution varies linearly. So the systems are completely different. And of course, the QTOF has been the mainstay of all of this work, and it's virtually unlimited in terms of M over Z range. So if you are working with very, very, very large complexes, I would say this would be your first choice. It's a little bit limited in terms of MSMS possibilities. Most instruments can do collision-induced dissociation, others will have lasers or other possibilities, but usually they are not great in this aspect. You are limited just to one cycle of MSMS. Of course, you can couple QTOF instruments with ion mobility, and this gives an added flexibility. So basically, I would say these instruments have adequate resolution and mass accuracy. And I started in ATMS, of course, with a QTOF instrument. For the Orbitrap, the situation is a little bit different. You have, of course, better resolution, very high resolution, mass accuracy, easily exceeds what you have on here. And of course, you are limited only again to a cycle of MSMS, but you have more possibilities. The instrument is more amenable, not only ECB, but there are modified instruments that can do electron captive dissociation or even UVPD dissociation. These are experimental instruments that are installed maybe in 1 or 2 laboratories around the world, but they show a trend towards the future. FTICR is a completely different thing. So its ion optics is much similar to the QTOF, so this instrument transmits very well. The M over Z range that is accessible is limited by the magnetic resonance analyzer. So I would say it's lower than what you can achieve on this one, but still you can go easily to analyze complexes in the 1 megadalton range, as has been demonstrated by Joe Lowe and others. One of the main advantages is that you actually have an unlimited MSMS depth. You can manipulate the ions in the gas phase. You can use all sorts of activation processes on collision, lasers, sorry, bird. I could spend all my time and even more talking about what you could do with all this, with all these methods. But all this has tremendous flexibility to your structural interrogation of molecules in the gas phase. And intrinsically, the detector has a resolution that exceeds anything else by 10 to 100-fold. But for some reason, we are not yet being able to extract and squeeze all this resolution. So what can we do? Now, one of the things, of course, is improving the solvation. And this shows the effect of different nebulizers. So these are the small capillaries where your sample is placed here, around 1 or 2 microliters, and then they fly by nano spray. If you look at this, you see that the shape of these tips, which are handmade and gold-coated, you can also use a metal wire inside if you like. Personally, I prefer the gold-coated, not only for safety, but it's usually more reliable, although it may not hold a stable spray for, say, as long as a wire in contact with the solution. But the shape actually influences not only resolution, but also the stability of your complexes. This is a tetrameric protein. You can see in this case, you have a slightly lower resolution and you start to see monomers to carp up. So something that you would not think as being so critical actually has a very important effect. And optimizing all this is quite important for obtaining the best possible results. Even more interesting is that you actually use different sources on the exact same instrument on the exact same conditions of tuning. So in this case, if you compare the electrospray ionization with nano-electrospray online, so this is offline, this is online, you can see the charge state distributions here, and you see something is starting to come up. But as you lower the flow and reduce the size of the droplets, you see much better resolution. So exact same instrument, no modifications, and I would say minimal tuning to obtain this. So we expect to keep increasing the development of the source so that we can get the best possible desolvation. This is quite critical for a good result. I mean, the highest resolving analyzer in the world cannot do much of this. Better with this, and of course much better in the future as we develop this approach. So one of the things that revolutionized the field of FTICR is the introduction of a new magnetic resonance cell that was invented by Evgeny Nikolaev, also a member of our consortium. So this was invented and came to the world, I would say, in maybe between 2008 2012, until it became a commercial product and started to be integrated in the in this mass spectrometry. So I would say in the previous 20 years, everyone used conventional cells and for the main situations, because there's presently just one manufacturer of this type of instruments in the world. Most people would use the so-called infinity cell. So the infinity cell is also a magnetic resonance cell, and you measure the transient of created by the image current generated by all ions inside the cell in a magnetic field, while they are trapped also by an electric field. And previous cells would have a relatively sharp decay as you would increase the length of the acquisition. So acquisition time influences resolution. The longer the acquisition time, the more resolution you can actually have. So previous cells, you could hardly go, say, to this length. And you need about 3 minutes to acquire a spectrum where you have something like 24 million resolution at a relatively moderate field. So the trick for the cell is actually carefully controlling the position of the ions in the cell and carefully controlling the electric field, both for excitation and the detection. So you can actually play with your ions much better than in previous cells. Of course, there are other designs, and it's possible that in the foreseeable future, there are further developments that allow us to reach even more and more resolution. So maybe one day with this type of instruments, we can actually see all the isotope loads of even the hardest, highly modified, largest molecules. Of course, one of the goals is to obtain isotopic resolution. This is something we can do presently, but mainly for relatively small proteins, either individual ones. So despite the look of the spectrum, all this is isotopically resolved. So we can calculate the masses very accurately for a protein of around less than 20,000 Daltons. You can calculate an accuracy that is in the very low Dalton range. So with this kind of accuracy, we actually monitor the reversible modification of this model protein with different chemical compounds, with the idea to create a soluble and targeted drug delivery system. For a small complex, which is about 32,000, we also have isotopic distribution on this kind of systems. Resolution here is important. You can see the charge state distributions for the dimeric form. But in this case, resolution is very important to decide what kind of metal we have, because we did the structure of this dimer by X-ray crystallography, and of course we have metal analysis, but it's still difficult to say what kind of metal we have, because the differences were not so obvious. So actually weighting the molecule unequivocally told us that this dimer actually has 2 zinc ions and no other ions. So what is our roadmap for future developments as part of Instruct? We would like, of course, to continue contributing with this technology, because we believe in the future the advantages of using magnetic resonance for native mass spectrometry will bear its fruit in terms of extreme resolution that is not presently accessible by any other detector. So The main developments will be, of course, in improving the source so that you can get the best possible desolvation, but also playing with the pressures at different areas of the instrument. So our instrument, the guarantee is just over recently, so now we can start playing with it and eventually make some modifications to better control the pressure on the system. So the other way, of course, is to increase resolution by non-hardware means. What I mean is that there are some techniques basically to process the Fourier transform, so-called absorption mode, that doubles the resolution and increases dynamic range, or resort to quadrupolar detection, which actually has the potential to double the resolution. These two approaches in FTICR have been used in many situations, mostly to obtain the same type of resolution in half time, so to obtain a faster duty cycle. On rare occasions, this has been used to extend as much as possible the resolution we can have. And the other one, of course, is to take advantage of all these isolation modes that are also unique to magnetic resonance. And this would allow us, say, to take a complex in the gas phase, dissociate the complex, then isolate one of the proteins, do MSMS of the isolated protein in the gas phase to identify PTMs or something that is bound in a more stable form. So the possibilities are endless. Or even using a technique that is two-dimensional MSMS, which allows us to do all ion fragmentation, and of course do the assignment in equivocal assignment of all fragment ions to the precursor ions. Presently, this is only done in 2 of the laboratories of our consortium. Of course, there is a last resource, and that is increased magnetic field. Magnetic field does have, despite being linear, the effect on resolution is present, and there are other effects that are also quite relevant, especially the dynamic range, and this increases the quadratic of the with the magnetic field. So for some applications, I would say the magnetic field increase will have desirable effects, but it's something that we are considering only after taking advantage of all the other possibilities. Because even at 7 Tesla, we have the potential to achieve very, very high resolutions, as has been demonstrated of more than 20 billions. So this is my team at the Faculty of Health Sciences. Most of the native work is actually done by a small part of the group. That means Mark, Marianne, and Margarida did most of her work on this. And we also integrate several international initiatives like the Consortium for Top-Down Proteomics. And if you want to know more about Fourier transform mass spectrometry, magnetic resonance mass spectrometry, in general, extreme resolution mass spectrometry, please join us in Lisbon for the European Fourier transform mass spectrometry workshop that has been postponed twice due to COVID, but I'm very confident that on the 3rd attempt, we will be able to host it. Thank you very much for your attention, and please let me share with me your questions, doubts, and anything else.
Okay, Carlos, thank you very much. I have some comments and one question here, but maybe I will start with 2 questions. Okay, I was impressed with your first picture where you showed the resolution of related with our padrões de descobrimentos, our statue about the discoveries, Portuguese discoveries, where you showed the increase on resolution on what you see with these various instruments. Of course, it was not the only slide that impressed me, but to start up, I think that was a very, very good approach. However, there is a difficulty here, which for most of our structural biologists, that I guess many people will have on their minds at this moment, which is what you mean by resolution is not transposable for what we mean by resolution in X-ray crystallography or cryo-EM. Yes. And maybe you could explain that, and then I will come up with the questions presented by some participants, because I think these you increase, you quote numbers of millions, of thousands, and for us resolution is going to a lower limit of what we can see. So there is here difficulty on words and meaning of words that maybe you could explain to the audience, because this could be helpful.
Yes, it's exactly the opposite. For atomic coordinates, the lower the better. For us, the higher the better. For us, mass spectrometry, resolution basically is stability to distinguish different things. We need to be able to distinguish if we want to have charge state distributions or isotopic distributions, and this is how we can accurately calculate mass. So when you have a resolved peak, like in a chromatogram, for example, size exclusion chromatogram, so you can quantify that resolution. You use basically the 8 of the peak divided by the width of the peak at half height. So the taller the peak and the narrower it is, the higher the resolution. So this is a quantitative measure of resolution when your peaks in the mass spectrum or in the chromatogram are baseline resolved. So that is why you have this increasing and increasing
number. Yeah, but you are not aiming at having to see individual molecules as a whole. You're aiming at differentiate between what you have in your sample in terms of the various components of the sample. Is that correct?
Yes, I illustrated the effect of resolutions on 2 different situations. One on the analysis of small molecules, and this has
here the audience is mainly large molecules, yes. I suppose.
And because the problems are completely different, but they take advantage of high resolution. So for large molecules, it's always the same molecule, but in different charge states.
Okay. So it's complementary to the other techniques.
So you use this information to calculate the mass. And if you have high enough resolution, you start to see the isotopologue distribution of that molecule. You start to see what kind of isotopologues compound are the components of this molecule. So for proteins, you really need an outstanding resolution to see more than the carbon, the distribution of carbon, and this is usually what we can see. And I would be very happy to be able to see that at larger complexes. For example, we can see it relatively easy with small complexes. I would say 50,000, 90,000, around 100,000 Daltons. We can see this without much effort. But if we want something above that, say 1 million, we see charge states, so we are still struggling to get this kind of resolution. And I think the full isotopic resolution for a protein has only been achieved with denatured forms and low molecular mass proteins. So we still have a long way to go, but the potential for resolution is there.
Okay, now I'm coming to the questions from the participants. We have here José Maria Carrazos that says, very nice talk, Carlos. Any plans to work with complexes in the future, larger size? Well, related with what I...
Definitely. Of course, we are starting small and we want to go up the ladder. I think up to 1 million Daltons, we should be able to see it in a much better resolution that is presently possible today with other instruments. So that would be our target. So at the moment, we are mainly working, of course, with some model proteins and others that we find interesting for our research. But yes, of course, the larger the better. And of course, we would like to combine this information and to integrate with other structural studies. I think native mass spectrometry at this resolution matches very well what can be done with other techniques, especially CryoEM.
Okay.
You can use this as a screening to say, I have complexes. The complexes are homogeneous. They have this mass, so most likely this is the stoichiometry. And you can have hints on the topology of the complex long before you go to CryoEM.
We hope to see more of that within Instruct. So the last question is from Jan Janis that asks, how do you foresee combination of ion mobility and MRMS in the future for native MS?
Nice to meet you here, Jan, and also Jose Maria. For Jan, yes, I do foresee the combination of this, and I'm sure he also does, because he also works on the same line. And I think these two things would be our first upgrades in our wish list for our instrument. That and 2XR to double the resolution. Because if we have high mobility, of course, we have another separation, and we can explore other things like conformations, and will give us a huge benefit. As far as I know, there are maybe 2 or 3 papers on this on FTICR. There is one approach that is using the ion mobility that is mounted by Bruker on their instruments on the timsTOF. But the timsTOF is a QTOF, and the QTOF is an extremely fast instrument. So the time scale of operation, of course, is much better. But there has been a couple of papers where this kind of ion mobility has been used, and the results were encouraging. There are also some manufacturers that produce other options for ion mobility, for example, external atmospheric pressure ion mobility, and these are 2 things that would definitely be worth to try, because they don't add much in terms of cost or effort, but they do add a lot in terms of functionality. Okay,
thank you very much, Carlos. And we're moving to the last speaker of today's session. Thank you. Well, Jan says also thank you, Carlos. See you in April. I don't know, maybe you'll meet in April. Okay, now we will have Pedro Matias from ITQB, University of Lisbon, who will tell you about what we have in Portugal at the moment regarding CryoEM and what are our future possibilities, we hope. So the title of his talk is Catching Up with the CryoEM Resolution Revolution.
Thank you, Maria Arménia, and thank you for the invitation to give this presentation in this meeting. It's a bit off topic in a sense, because I'm not really going to present any results, but this is an important step that we need to do in Portugal in order to really be up to par with the other developed countries in the world. So I am going to give you this presentation, but I would like to also to acknowledge my colleagues in this endeavor from different institutions, from ITQB and also from the IGC, I3S in Porto, University of Minho, and the INL in Braga. So all this began in 2017 when we learned of the award of the Nobel Prize in Chemistry to these 3 outstanding scientists. We knew that this was really going a game changer in terms of structural biology and other applications. But of course, we also knew that we did not have any instrument in Portugal with those capabilities at the time. And in terms of health science research in Portugal, there are groups that work at different levels, from small molecules to proteins, microorganisms, but cells and tissues. And these in different parts of in different universities and institutions throughout the country. So we were aiming, because we know that this is to get a CryoEM instrument is a huge investment. We were aiming for to raise the largest possible critical mass of interested users. So the idea was to go for an instrument which could be as flexible as possible. And to set up the ground for that, we carried out the research in 2017 to try to identify the main fields of research of the scientists that would be interested in the setup of CryoEM facility in Portugal. And here you can see that the core work is in the structural biology, cell biology, and the biochemistry, although there are a few other applications which are also significant, but they are very distributed among very specific applications. And also in terms of the techniques that you can use with cryo-EM, the main ones were cryo-EM tomography and single particle analysis. And of course, the ideal was to get an instrument or to go look for an instrument that could serve all of these, but technically it might not be entirely possible. So we wanted to get to cover as many as possible. And it's not here, but also in 2017, 2018, a new field emerged called microelectron diffraction, which is very interesting because you can use very, very small crystals to get diffraction, electron diffraction patterns in the microscope, and then you can also solve their structures. And this is useful not just for macromolecular crystallography, but also for small molecule crystallography, because it can deal with very tiny crystals of organic molecules, which are harder to grow larger for standard crystallography applications. So this was also something to be included in the portfolio of applications of an instrument to be set up in Portugal. So it's not like there were no electron microscopes in Portugal. The problem was that most of them were very old. And there was even a microscope at the INL in Braga that could do basic cryo-EM, but a very limited way. And they were all below the 200 kilovolt threshold that normally is used for cryo-EM. And so, of course, you heard from Hugo that this is not strictly true, what I'm going to say, but because there are no state-of-the-art microscopes in Portugal, it is very difficult for us to get the preliminary data that we can use to meet the access requirements at these centers. And I'm talking particularly about the 300 kilovolt instruments, the Titan Cryos and the like, which are presently available in several countries in Europe, and they are available through Instruct. But we need preliminary data to show that our samples are enough good quality. So you need to have devices for vitrifying your samples, and then you need to have a way of checking the quality of the samples, that they are good enough, because these sites are available, but they're very competitive. And if your case is not strong enough, you will not get time there. So this was also a key driving force for setting up a national center for cryo-EM in Portugal. Of course, through Instruct, we can also access intermediate services that can give us this information, but it's not the same thing as having a national facility, because it will be much more flexible than having to constantly rely on Instruct applications to get access to these facilities. So we received support from many public and private higher education institutions and research institutions spread out throughout the country. Also, a couple public hospitals and some pharmaceutical and biotech companies also showed interest in supporting and using such a facility once it became available. And so in 2018, we set up our plans to propose a national cryo-EM facility equipped with a 200 kilovolt screening microscope and a 300 kilovolt state-of-the-art microscope. The 300 kilovolt fell by the wayside when we started analyzing the costs of maintenance, because these instruments are not only they are very expensive, but they're also very costly to maintain. And then the cost of maintenance has to be covered from user fees, and then the users have to raise the funds from their own grants to cover it. So we decided that the 200 kilovolt would be already good enough for most applications, and it would already provide the means for the Portuguese researchers to access the higher-end state-of-the-art microscopes in Europe. So this facility would support the industry to allow innovations in the field of pharmaceuticals. Another important aspect at the time when we had the contact with some companies was that the cryo-EM particularly in the field of cell imaging and similar applications, was very important for them to meet the regulatory regulations when they wanted to certify new drugs in the market. And having this facility in Portugal would facilitate their life tremendously. Also, in terms of the hospitals, the view was such a device or such an instrument would, in certain cases, improve the diagnostic capabilities. And finally, of course, in terms of academia, it would really allow the Portuguese scientific community to develop projects in the field of health sciences that would not be possible otherwise. And I'll just give you a few examples. So of course, then there would be a virtuous cycle where all of this knowledge would cycle back into the industry and the health services. So one of the projects was and is being developed here at ITQB by my colleague Margaret Archer. She's also working in anti-tuberculosis drugs. And the idea is to characterize mycobacterial membrane enzymes that are involved in biosynthesis of the cell wall components. And so they have at least one structure of such an enzyme, and they had to collect data in the United States within a collaboration because it was not possible to do it here in Portugal. And then in Porto, my colleague Sandra Rivaro is interested in protein misfolding. So she's studying toxic fibers that form from disease-related proteins. And the CryoEM also would allow her very much to study this problem and to really develop knowledge in this area. Then in Coimbra, Ana Luisa Carvalho is interested in tissue structure in neuropsychiatric disorders, and she would also be interesting to study the tissues using cryo-electron microscopy for imaging. And closer next door to us at the IGC, Maria João Mourinho is studying the influenza A virus. And this is something actually that I didn't know, that there are two kinds of influenza A. One is the spherical and the other is the filamentous. And her goal is to study the filamentous virus and to understand the life cycle to improve treatment against the disease. And so where to place the microscope? The obvious, after a bit of consulting, the obvious decision was the INL. The INL is the International Iberian Nanotechnology Laboratory. It is located in Braga. It was founded, if not mistaken, in 2009. It was a joint initiative between the Portuguese and the Spanish government. So it has extraterritorial statute, but it is specialized in electron microscopy, mainly in the material sciences, although they do have a small biology research group. But they have already infrastructure, experience in the use and maintenance of microscopes, and also they have partnership with manufacturers of these microscopes. And very importantly, the inset here shows you an empty room where a new microscope could be installed, because these microscopes have to be placed in special rooms protected against electromagnetic fields and vibrations and so on, so that the images that we get are the best possible. And so the setup of this kind of facility in Portugal would be tremendous at different levels. And here you have a list of different aspects that we wrote down in our proposal to support the creation of the facility. It was realized that this would be a national level effort, even though the instrument would be located in the north. It would affect groups all over Portugal. And so to try to get funding for this, because of the way the funding structure was set up, when you want to use EU funds, which is the obvious choice for this kind of facility. We had meetings with the regional development commissions, which are the organizations that control the award of these funds for research and other purposes. But we had a problem because it was difficult to find a mechanism to really build or to really fund this kind of initiative. So in the end, we set up the proposal for a network, which we christened the National Advanced Electron Microscopy Network for Health and Life Sciences. And we were able to get this network integrated into the National Infrastructure Roadmap. And this was the only way that we could apply for funding under different funding programs that would eventually become available. And as you can see here in the map, we have the nodes spread out throughout the country. The central node would be in Braga, but then we would have throughout mainland Portugal nodes in different parts of the country. And we were able to obtain funding for the 200 kV CryoEM to be installed at the INL next year. The remaining nodes will be implemented as new funding opportunities become available. And right now there's ongoing call for the renewal of the National Infrastructure Roadmap. So we are going to submit a revised application for our network, and we are going also to include some additional nodes from institutions, mainly in the Lisbon area, that manifested their interest to be included as new nodes. But okay, now we have a network which is kind of still its infancy. We don't even have a CryoEM, but then we need to now to use it best. So in 2018, we applied for a EU project on the Twinning Horizon 2020 program. The first call was not funded, the first try was not funded, but we did get it on the second try, which was good news and bad news. The good news was that we got it. The bad news was that from the first try in 2018 to 2019, the budget decreased from 1 million euros to 800,000 euros. So we lost 200,000 euros in funding just from not being able to get it on the first try. So the mission is to bring the CryoEM knowledge to ITQB, and from ITQB then to spread out the knowledge through the structural biology community in Portugal. And the main activities envisaged were and are workshops with hands-on experience. Unfortunately, the COVID-19 pandemic really put a large wrinkle into that, because we had 3 workshops planned. We were able to do the first fully hands-on in September 2019 at ITQB, and then we had 2 other workshops planned for 2020. And because of the pandemic and the restrictions that ensued, travel was severely restricted, and of course, we could not move, we could not go to the other countries, and so these had to be restructured. And although we were able to circumvent the problems to some extent, it's not going to be the same thing, unfortunately. The second level was to send early career researchers from ITQB to the partner labs. We already have a few visits, even despite the COVID-19, they have taken place. We organized a series of seminars by world-leading experts in the field. Among others, we were, of course, after September, they had to be virtual also, but we had the honor of also welcoming Professor Richard Henderson, one of the Nobel Prize winners, as one of our speakers. And in addition, because these projects also deal with development for early career researchers, we also included a few soft skills development courses in our project, in our activities program. Most of them have been also done virtually. And finally, outreach activities for the general public, where we divulge not only our own research, but also the importance of CryoEM in general. And just to finalize, just to show you our consortium, our partners. So we have, we were fortunate to build a strong consortium in 3 different edges of triangle, or vertices actually. So on one side we have the CSIC headed by José María Carazo, and also with Carlos Oscar Sorzano as an important team member, where they have acquired a tremendous knowledge in imaging processing methods for electron microscopy. At the University of Helsinki, we have Sandra Rivaro, who is specialist in the single particle analysis of macromolecular complexes and nanoparticles. And also in Israel, we were able to connect with the group in the Weizmann Institute of Science, where our PIs are Michael Elbaum and Sharon Wolf for cryo-EM tomography. And so just to finalize, like I said, we are hoping to get our first cryo-EM microscope operational next year at the INL in Braga, and it will be a momentous occasion for us, and hopefully not too long after that we will be able to really start implementing the different regional nodes in earnest. Thank you for your attention.
Okay, Pedro, thank you very much. You set up the picture of what we have in Portugal, and we are slowly building up, and hopefully next year will be an important year. In the meantime, we are well connected with through Impact with the other Instruct centers, as you have indicated, and we have been profiting from that very strongly already in terms of research projects and developing research projects from Portuguese researchers, which is very important. And also maybe I could add from what you said that the new site of the Instruct Center in Spain, we will try to have a large amount of their time devoted to users, to help users on CryoEM, and thanks to José María Carazo and Carlos Oscar Sorzano, we will be very much benefiting from that, and therefore through not only Spain but other centers within Instruct having access to high resolution CryoEM, which is very good. So that's why we are advancing in Portugal. This is not a presentation related with structural results, but more on the situation of Portugal of CryoEM in Portugal. Okay, I don't know if there are any other comments. You have some comments in the but papo in the oh, I don't know how to say that in English. Anyway, I have it in Portuguese, but in I
think they all reply to the previous speakers.
Yeah, but nevertheless, thank you very much, Pedro. Thank you for all of those who worked with you to get all these two initiatives getting along. Especially Celia, which was your right-hand helper, I know, and also a lady from IGC, which is another area, but very important as well. And good luck for the future. And thank for all of those who participated in our Structure to Function webinar series organized by Instruct. And thank you, not last but not least, the hub for support, especially John, Dolan, and Pauline. Thank you very much, and see you again sometime for the next webinar series from Instruct. Thank you.